Hi everyone, so today we're going to go over RNA processing, and yeah, it's a really simple process. So essentially what we want to first know is that RNA processing, or in other words is post-transcriptional modification, occurs right after transcription but before translation. So it still happens in the nucleus. All it does is it turns the pre-mRNA strand that we got from transcription into mRNA, mature RNA. Right? And this process is mainly done to aid in stability as well as other things. So like I've written here, after transcription you have a pre-mRNA strand. To turn this into mature RNA, there are 3 specific processes that occur. And yet, like, these processes aid in stability, as well as the fact that pre-mRNA cannot directly be translated because it does contain unnecessary sections. There's sections that we have to cut out, as well as it also does lack the ability to kind of move about freely. It does lack the protective structures that it needs. So all of these things do aid in each of these, as well as stability. Well, so the first thing that happens, the first step that happens is that a 5-inch methyl cap is added. So let's actually rewrite this. Yeah, so a 5-inch methyl cap is added. 5-inch meaning it's added to the 5-inch end of the thing. So we can have a strand going from 3 to 5 as we know. So the methyl cap will be added here. So actually it's kind of, yeah, no, it's just right like that. That's that. So this would be our methyl cap, right? You don't need to know the composition or anything like that. You really just need to know what's added and why it's added. Well, so basically this methyl cap is a modified guanine nucleotide. It's added to the 5-inch end of the pre-mRNA, as we said. The kind of functions of this is that, one, it prevents enzymes from kind of breaking down the DNA, so it prevents enzymes from breaking, sorry, not DNA, mRNA, breaking the mRNA down. It prevents enzymes from doing this. Another thing it can be that, again, we said that it aided in stability, so it does help mRNA exit the nucleus and attach onto a ribosome, as well as the ribosome uses this to, essentially, a ribosome attaches onto the mRNA strand, so it needs signals to help it kind of identify where should it attach, like what should it attach to. So the methyl cap helps for identification. So we'll just write methyl cap helps identify helps in identification, let's just say. So yeah, you can take a screenshot of these notes. Yeah. So that is the first step. That's basically all you need to know. You don't need to know all 3 of these. I think one example should be fine, or one reason should be fine. So that is the first step. So let's just highlight this. So methyl cap is added to the 5-inch. So the second step of RNA processing is that there is something called poly-A tail, which is added to the 3-inch end. So you have the methyl cap added to the 5-inch end. Now you have a poly-A tail added to the 3-inch end. Now, what is a poly-A tail? Well, we know that poly means, is just a long chain of things. So we have a polypeptide, which is a long chain of amino acids. Poly just means there's a lot of something, right? So poly-A tail means there's a lot of adenine kind of joined together. So A represents the structure adenine, similar to how in DNA you have ATCG. Adenine is an amino acid, so there's quite a lot of poly-A tails added together. So there's usually around, like, I would say, like, anywhere between, like, 60 to, I'll say 300, maybe not 300, I think 60 to, like, 250 maybe, 60 to 250 adenine tails added, or adenine amino acids added to the 3-inch end. So essentially just a long chain of adenine nucleotides added to a 3-inch end. And why is this important? Well, why is this needed? But one reason is that, again, we can say that it protects mRNA from breaking down. Let's just say breaking down. Another reason can be that it has more protection, basically. Yeah, so we have more protection. Well, it's just the same thing. But another reason it can be it aids in stability, where it assists transport outside the nucleus, so it helps it exit the nucleus. So that's another thing. That's all you need to know. The third one gets a little bit more complex. This is what we call splicing. Now, splicing does sound a lot like slicing, right? So you can use that as a hint. Splicing, all it does is that specific sections of DNA are removed. Now, DNA contains coding and non-coding sections, right? Non-coding sections that don't code for the specific thing that you want, such as like the protein or whatnot. Coding sections do. What we call non-coding sections introns, right? We call non-coding sections introns, and we call coding sections exons. Essentially, this is more specifically intron splicing. What we are doing in splicing is removing the non-coding sections, because if it doesn't code for what we want, why do we need it? So this is called intron splicing, and this is done by spliceosomes. You don't need to know how exactly it's done, you just need to know that it's cut and whatnot. So essentially what happens is that introns are just spliced out. Don't say they're cut out, just say, to be more technical, kind of ensure you get good marks and whatnot, just say they're spliced out, sorry. So these are done by spliceosomes and whatnot. Now, why is it important? Again, only exons should really be translated, because they're the coding section. They contain the specific sequence that we want. Introns generally disrupt the amino acid sequence, so we don't really want that. And yeah, that is basically all that is there for post-transcriptional modification, RNA processing. That's basically what we need to know. You realistically just need to know the three processes were really simple to understand, as well as a couple of reasons why. But yeah, that is about it. If you have any questions, please feel free to ask, or visit our website or our YouTube channel. Thank you so much for watching.